All right, so here we go. First set of notes for human biology. So we're going to start off with scientific method. And I know that you guys have done scientific method probably every year within science, but we want to go through it just really quick because this is kind of how you guys are going to be set up in your labs. And we want to make sure that you guys are hitting each one of these steps and you kind of understand what these steps are asking for along the way. So as you're going through these and the little video, these notes are downloaded into Google Classroom as well, so you should be able to pull them up and look at them as you are going through your little assignment, which is scenarios for this one. We'll also be doing a little activity within class. Then we'll pretty much be done with scenarios as well as scientific method. Moving on to water at the end of the week. So this, like I said, real quick little section for you guys just to get you started here. So anything that you do within life, you guys actually use the scientific method whether you know it or not. So scientific method is just kind of a way to go through and solve a problem. So you might be home and hit a light switch and the lights don't come on. You don't sit there and go, oh, let me think about the steps of the scientific method in order to solve that particular problem. You don't go, oh, well, the lights don't come on. Could it be a fuse? Could it be this? Could it be that? I'm going to experiment. I'm going to do all this other stuff. You just kind of go through and do it. But anything that we do in terms of labs, you guys will be going through these particular steps. So any lab that we have or anything within this science field that we see, you guys will always be presented with a problem. What are you trying to solve? So this week's little activity that you guys will see, which effervescent tablet or which temperature causes the effervescent tablet to dissolve most quickly? Is it going to be the cold water, the warm water, or the hot water? And it's going to be an easy little experiment just so that you guys can start to see where these steps are seen within that particular lab. So once again, problem, you guys, what are you trying to solve? Go through, read your little scenario, read your lab. This is always given to you within an experiment. You guys need to create a hypothesis. Your hypothesis, if you are working with partners or collaborating within groups, does not have to be the same as everybody else in your group. You can still work together and collect data. This hypothesis is yours. What do you think is going to happen and why? It doesn't have to be correct. This is why you guys are testing it. So you should never go through, create a hypothesis, get to the end and go, oh no, my hypothesis was wrong. Go back, scribble it out, and create me a new hypothesis. You will never get marked wrong for having a wrong hypothesis. You'll justify it in your conclusion and you'll tell me about it. Yes, the hypothesis was correct. No, the hypothesis was not correct. This is why. So with that hypothesis, things that we're looking for and where you could lose points, you need to be bold. Never, ever put I think, never put I believe. Be bold with it. So when you do that, just cut those words out. Usually if you start a sentence, I think that this is going to happen. Cut out I think. This is going to happen. Be bold with it. It is a possible solution to the problem that you guys are given. It's an educated guess based on the information that you know from previous experiences. It does not have to be correct. You guys are going to see constants and variables within experiments as well. So constant is anything that is going to stay the same. So you'll have a couple different setups in this particular experiment. Even when you guys do supervisors activity, your setups, you're going to have cups of water, and you're going to be testing cold water, warm water, as well as hot water. And you're going to kind of have this control, and the constants are everything that stays the same. Same size cup, same amount of water, same type of tablet, same timing method. Everything that's going to stay the same between the experimental setups that you guys see. The variable is the one thing that is going to be tested for. So once again, when you guys are doing that supervisors, the variable that we're looking at cold water, we're looking at warm water, and we're looking at hot water. That's the one thing and only one thing that can change between those experiments. We don't want to test two different things. Well, let's test different temperatures of water and different amounts of water. Because then if something changes in an experiment, we don't know what caused it. Is it the different temperature, or is it because I put different amounts of water in each of the cups? So you can only test one variable at a time. Within those variables, we look at independent variable as well as your dependent variable. And you've probably heard of these before. We do some graphing. You guys will graph a lot of your results, which you can be done on the computer, or you can do them by hand, snap a picture of them, and send them in that way. Your independent variable is the one thing you are controlling in the experiment. What are you controlling in the experiment? That's always going to be on the x-axis. So are you controlling time? Are you controlling temperature? Whatever it may be, whatever you're setting those boundaries as. This is what I'm testing. I'm testing it every 5 seconds. well, then you're controlling the time on that. Your dependent variable are the results that you're obtaining. So it's dependent upon the information that we're putting on our independent variable, or our x-axis. So that will always go on the y-axis, those results, and they will depend upon what we have here. This is going to play a role on the effect of our data that we're collecting. The control is our norm. What do we normally have? So we always set up this basically control, and this is going to be used for comparison. So you guys will see some examples coming up soon with peat moss, and we might say, well, I usually don't grow plants with peat moss. That's my norm. Well, what happens when I add peat moss to different soils? How does it change things that I'm growing? Your experimental group is what you're testing, so you're adding different things to it so that we can get data. And then one of the last steps that you guys have, as you are collecting data, you want to create a chart. And from that chart, you can create a graph. So your data chart is going to be a way to organize your information. You guys will graph, you guys will chart, you guys can create a table. It isn't organized, clear, it needs to be logical, it needs to be neat, it needs to be labeled. So what are those columns within that data table representing? And you guys are always looking for two types of data. And a lot of times we forget the second type. We're always really good with quantitative data. Oh, we're measuring plants, and the first plant grew 5 centimeters, and the second plant grew 8 centimeters, and the next plant grew 10 centimeters. We always collect quantitative data, quantity, numbers, and that's what we're looking at is actual measurements with units. We need to make sure we include units, centimeters, feet, miles, inches, days, weeks, years, seconds, minutes, whatever it may be. You need to make sure that you're including units with that. The one that we kind of forget about is that qualitative data. Quality, which means it's more descriptive. The first plant is more green. The second plant might be a little bit more yellow in color. A third plant has wilted leaves. Tell me about it. What does it look like? It's non-numerical, but we want to look at colors. We want to look at sizes, maybe textures, maybe the way it sounds. Is one bubbling more? Is one fizzing more? Whatever it may be. Maybe one's got a weird smell to it. Whatever it is. But you need to put descriptive data. So we're going to be looking for both for qualitative as well as quantitative data. You guys will see some stuff underneath Google Classroom too that's extra information for you. And part of that is going to be how to set up a good graph. And like I said, we do a lot of graphing within this class because we collect a lot of data. And I'm going to go through some points on where you guys are getting your points when you're making the graphs and what we're looking for. So once again, if we're setting up a little graph, here's my X and Y axis. Remember that X axis is the horizontal axis and my Y axis is up and down. X axis is your independent variable, what you're controlling. Your Y axis is your dependent variable. It is the results of the experiment. So what we're looking at, and when you guys get to your scenarios, what you guys are going to see, the first one's kind of a little practice one for you. So your little practice one that you guys are going to see, let me go through this first, looks like this. And this talks about the peat moss scenario. So if you guys don't know what peat moss is kind of almost like a mulchy-like material, and it gets added to soil to help maintain the moisture within soil. So if you're planting trees or plants or something like that, and it's more of a porous soil, you want to add peat moss to it so that it keeps the soil a little bit more moist for the plants to grow in. So your little scenario that you're going to go through, Norm wanted to know if adding peat moss to sand would affect the ability to hold water. So he puts 200 milliliters, known as the units, of pure sand into container A. He put a mixture of 80% sand and 20% peat moss into container B. Into container C, he places a 40-60 of sand and peat moss. And then we also have container D. So you guys will see all of these down here. So container D has 100% of that particular sand. He adds water to the container, measures the amount of water that the contents would absorb. He dried the sand and the peat moss, repeated the experiment 5 times. That's the next thing. We want multiple trials on that because we want to make sure that the validity is good. If we just run one trial and maybe something is messed up, well, then how do we believe those numbers? If we run multiple trials and the numbers are pretty consistent, we average them out, then we know we're a little bit more valid. And you guys will see that rather than running multiple trials, you guys might be collecting data from other lab groups and then averaging that information out to make sure that it's more valid. So there's our little data chart that we're given, and it's going to ask you the same things that we just kind of looked at. So thinking about that, first thing we're going to do is think of a problem. So if we're looking at it, let's look at this. So our problem, and it kind of goes right out and tells you about Norm. He wants to know if adding peat moss to the sand would affect the ability to hold water. That's it. It's always given to you guys in the experiment. So does adding peat moss to sand influence ability to hold water? Does it need to be that word for word? Absolutely not, but it should have something in there. And what else you guys are going to notice is that I don't really have any personal pronouns in here. There shouldn't be within your lab. Okay, keep it neutral here. No personal pronouns. I think that this is going to happen. I believe this. I this. He this. We this. Keep it all out of there. So now we want to come up with a hypothesis. So we're not really given Norm's hypothesis. Come up with one of your own. It's an educated guess. What do you know about peat moss? What do you think is going to happen in here without looking at your data, and come up with your own? So I say the more peat moss, the more water holding capacity it may have, because, and I could say peat moss has great water holding capacity for plants to maintain moisture, whatever it may be. Okay, so if it's not in the scenario, you're making one up of your own. So let me read this. Oh, let me come up with a little hypothesis. This is kind of like my experiment that I'm doing, but Norm's doing it for us. Within this experimental setup that Norm has set up, we have constants and we have variables. Constants, always more than one, multiple. When we go through this, what are my constants? I want to make sure that containers A, B, C, and D, they need to be the same size. They should be the same shape. I don't want to have one that's taller and one that's more flat, because that's going to change surface area on it. The amount of water that they're getting, I want to make sure that they all get the same amount of water. They should all be within the same temperature. They should all be in the same location of the sun. The fact that we have repeated 5 trials for all of them, they are all going to be the same. So you want to have multiple constants. As you guys are going through your scenarios, if you just have 1 or 2, it's going to lose points on that. You guys should have multiple constants and what's staying the same throughout that experiment. Variable is the one thing that we're testing for, but we're not just filling in that one variable. I want to know the independent and dependent variable. What will your x-axis look like? What will that y-axis look like? So my independent variable, what I'm controlling how much sand and peat moss is going into each one of those containers. So here I'm saying, oh, this container has this amount, this container has this amount, this container has this amount, and so on. My dependent variable, I'm testing to see how much water does it actually hold. That's what my data table there says. So it's my water holding capacity. But notice that I also have it in milliliters. You want to make sure that you are putting that in milliliters. Okay, so you'll see it down here on this particular one, even though it's not listed here. What is my control? What is my, my norm that I have? The norm that you have is going to be container A. And I say that it's container A is because it's 100% sand. That's what's normally within that area. And Norm is testing by adding peat moss. So if I have a normal, this 100% sand, that's normally what I'm going to find there. And then if I start adding peat moss, those are going to be my experimental groups. Containers B, C, and D. Okay, how does it change once I start adding that peat moss to it? So one control and my experimental setups that I have there. I don't need to create a data chart here because it's already been created for me. So I'm going to set up a little graph and label the X and Y axis based on the data. I don't want you guys to plot points. I just want to know, hey, here's my little graph. This is what my X axis is going to have. This is what my Y axis is going to have. So we just said my independent variable goes along that X axis, and that's my percent composition of mixture. Okay, so it's percent composition of, if there's units there, I would put units, but we just have percent. For my y-axis, my dependent, it's dependent upon how much peat moss I have in. That is going to tell me how much water that I'm holding. So that's what you guys will see here. So it's my water holding capacity. It's also what my data table is labeled at. Notice that all my trial numbers are listed there. And everything's kind of filled in. So whoever's looking at your data knows exactly what they're looking at. It's listed in milliliters, so I'll make sure that those units are there. Remember, your y-axis is just the results of what you have on that x-axis. So you guys are going to see 5 more little scenarios like this. You guys are kind of filling them in the same way. When you get down to that graph setup, once again, I do not want you guys to plot points. You guys are just kind of going through and telling me what it may look like X and Y axis on that particular one. as you guys go through, or if you need to create like a little data table with the information that you have, you guys can go ahead and do that. We'll also be working on a little scenario in class, depending upon whether you guys are A group, B group, or even that C group will get you in a kind of a virtual meetup with some of the probably the smaller groupings that I have within class, so it'll be a little easier for you guys. Okay, so once again, these are on Google Classroom for you guys. Keep yourself organized. Maybe download some of the notes and start putting them into certain files that you guys have easier access to, especially after the end of the school year where you may not have access to Google Classroom. Also check out extra supplemental information. It'll be a different tab on the side, on the left-hand side of Google Classroom, and you'll see a little video on graphing, excuse me, on graphing and the points that we're looking for when you guys create a graph and what you need in order to obtain all the points that you have there. Okay, so if you have any questions, please don't hesitate to contact us, and we'll go through this a little bit more with you, and we'll do a little experiment in class as well. All right, thanks.